So thank you very much, Sonia, for the introduction, and thank you very much to Sete also for the invitation to talk with you today. So I'm here to tell you about the BDNF and TrkB, which are two very important molecules that we are hopefully going to learn a lot about today, and how they are key in the context of psychedelic-induced neuroplasticity and their potential therapeutic effects. So let me also say that I haven't gone very deep into preparing an introduction, like a solid introduction for psychedelics, this being a psychedelic research organization. But as we have discussed already with the organizers, if you have like any questions, you want a bit more background, I'm happy to discuss any questions you may have during the Q&A session. Okay, let's go for it. So first of all, let me introduce the players we're going to be knowing more today about. BDNF and TrkB. They are two important molecules that are at the core of the antidepressant action and neuroplasticity in the brain, both in the early stages of development and also during adulthood. They are also very important in the mechanisms of action for conventional antidepressants and ketamine, for their plasticity-inducing effects and also their therapeutic effects. We are here today to talk about psychedelics. I will not talk too much about antidepressants and rapid-acting antidepressants like ketamine, but please do let me know if you would like to discuss a bit more of that. So you may have heard that conventional antidepressants, such as fluoxetine, the commercial name that many of you may know and may even have used is Prozac, have been proposed to work by blocking the serotonin reuptake inhibitors, okay, at the synaptic cleft, which in turn would, in principle, increase the serotonin concentration in synapses, and that is what has been classically supposed to increase all of the BDNF or brain-derived neurotrophic factor release that then would activate TrkB and lead to all of the therapeutic and plasticity-inducing effects of these drugs. Then there is also ketamine, which is a bit unconventional when compared to more classical antidepressants like the SSRIs, that has been proposed to block to work through the blockade of AMPA receptors, which is a receptor in the membranes of neurons, and through there, a complex cascade of events would induce this upregulation of BDNF release and then binding to its receptor that would be TrkB. Okay. Thing here that I wanted to highlight is that both conventional antidepressants and ketamine, in their classic mechanisms of action, have BDNF and TrkB at their very core. And you can see this in any number of different models and schemes that you can find online. They're always there, usually at the center. But what has been conventionally proposed is that these effects were promoted indirectly. Now, a few years ago, we published from Eero Castren's lab, where I have been developing my PhD work. That is the one that I'm presenting today, later on in this presentation. We published that antidepressants can actually promote plasticity and their therapeutic effects by directly binding to the TrkB receptor. And they do so in this kind of cross-shaped conformation that two TrkB monomers would adopt. That would be like in the middle of the membrane, in what's called the transmembrane domain. And they stabilize them in a way that the receptor can become active by the action of BDNF for a longer time. And this is the way that we propose the plasticity-inducing and therapeutic effects of antidepressants would take place. That fluoxetine, among many other pharmacologically diverse antidepressants, would bind to this receptor, TrkB, and promote the action of its endogenous ligand, that's BDNF. However, what we appreciated in this paper already in this work was that these drugs do seem to bind with a very low affinity, or rather low affinity, for TrkB. We think that this low affinity is physiologically relevant, and it may actually explain that you need several weeks of chronic treatment, daily treatment with these drugs, with conventional antidepressants such as fluoxetine, so that you reach enough concentration in the brain so that the drugs can facilitate this action on TrkB. In terms of the rapid action of ketamine, we also observed that ketamine is a rather low affinity binder of TrkB, but it is also true that ketamine seems to achieve the concentrations in the brain required for the fast-acting effects to TrkB already with a single dose. That's what ketamine has been characterized for when compared to other more conventional antidepressants. Okay, but again, we came here today to talk about psychedelics. And for that, there is right now a discussion going on what is really a psychedelic, but without going too much in detail into this, I will just say that for the purpose of my presentation today, I will only consider psychedelics those hallucinogenic drugs that are what is called classical serotonergic hallucinogens or psychedelics that act through the serotonin 2A receptor, that is this 5-HT2A that you see, and they activate it to mediate all of their hallucinogenic responses. Chemically, there are mainly three groups that we can classify psychedelics in. Those are tryptamines, for example, psilocybin and its active metabolite in the brain, that is psilocin or DMT. They come from the magic mushrooms as their active component, and also DMT for the Amazonian brew ayahuasca. That would be the dimethyltryptamine, DMT. Then we also have the phenethylamines, like mescaline here, that is isolated from the peyote cactus, that is also, of course, hallucinogenic. And finally, we have the lysergamides, such as famous LSD. I don't think that it needs an introduction, as if you've been alive ever since the 60s or later, you will probably have heard at least once of this drug. Otherwise, I bet you wouldn't be here today. So for this presentation and for the research that I will be presenting, I'm going to focus in the two psychedelics that are showing the most promise in the clinic, at least so far. And that will be psilocybin and its active metabolite psilocin, and then LSD. Right, so why psychedelics? Well, because you may have heard that they seem to hold potential for the treatment of different mood disorders. And there have been several high-profile publications in the last few years. You may know or may have heard that psychedelics are still actually classified as illicit drugs in many countries, in Western countries in particular, and that it's been very difficult to access them both for recreational or therapeutic use, but also for research. This means that psychedelic research has had some kind of not completely stopped, but has definitely been severely affected for the past 5 decades or so, that it was very challenging to do research with these drugs. It is still pretty challenging for us as scientists to access these drugs and work with them, but I must admit that nowadays, now that there is some evidence that they may have some potential in the clinic, regulations are somehow easing up for research purposes, and luckily more labs are able to access these drugs for research purposes to learn more about their potential. So, okay, about that, we have had a lot of focus lately on psilocybin, the active compound of magic mushrooms, that has some potential for the treatment of depression, and this trial with psilocybin versus another conventional antidepressant, escitalopram, for the treatment of depression. You can see that already with one dose of psilocybin together with psychotherapy, we have a drop in the symptoms of depression very early on that can persist for several weeks. This also seems to be the case for rapid-acting for conventional antidepressants that usually would be slow-acting. So it's a bit surprising that they started acting so fast after the dosing started. But again, I remind everyone that these are data, the one that they are going to be seeing in humans in the clinic at the moment. That is always in a very controlled clinical context where there is psychotherapists that will give support, and of course, very close supervision by trained professionals, psychiatrists. Okay, so there we see that psilocybin would, at the very least, have equivalent therapeutic potential as one very validated and commonly used antidepressant such as escitalopram. However, and this is a very big point for the presentation and the discussion we'll be having today, it's also true that the hallucinogenic potential of these drugs is limiting their widespread application. And this is because there is a concern based on some maybe anecdotal, but some evidence that patients with a family history of bipolar disorder or schizophrenia may develop an irreversible psychotic event if they are treated with psychedelics. This is obviously a very important concern and has led to many patients being excluded from participating in this clinical trial, despite in many cases, of course, they could benefit from the rapid-acting antidepressant and long-lasting antidepressant effects of psychedelics. It would be much worse if we trigger, for example, a schizophrenia or a bipolar disorder coming out of that. So this is why it's important to see the potential that these drugs have, but we also must keep in mind that these drugs must be better known and studied before they can reach wider applications, because there might be risks involved. There is also the concern that these drugs may trigger the HPPD, also known as hallucinogenic persistent perceptual disorder, which basically consists on after even a single dose with psychedelics, usually in a recreational environment. I'm not aware of any kind of situation that has arisen from a clinical trial so far, but in recreational use has been reported that for some people with a single psychedelic use, you could see some kind of a hallucinogenic-like response long after the psychedelic is not there. So perhaps like months, even years after that dose, you may have like some kind of a hallucinogenic perceptual change in the way that you perceive the world, sometimes visual, sometimes auditive, that can be either just like for a short time or can become chronic, which obviously becomes a very big problem for the people affected. So all in all, let's keep in mind these drugs have a lot of potential on one hand, but we should also get to know them better before they start seeing widespread application, because they may have some risks. So this has also led to a very hot debate in the field that is whether the hallucinogenic effects of psychedelics are required for their therapeutic outcome. And there are different positions at the moment, both partially based on evidence, although evidence is heavily lacking on both sides. There is, of course, the side, and we will learn more about this later, that proposes that psychedelics are therapeutic not because of the hallucinogenic effects, but rather because of other kind of effects, such as plasticity, which again we'll see more very soon. And others that argue that the hallucinogenic or this kind of psychedelic experience is required for a therapeutic outcome. And indeed, it's true that if you go and get to see what patients are discussing after these modern clinical trials, in many cases, these kind of more mystical experiences that they may have during the psychedelic dosing sessions may actually help them get better or get other perspectives from the problems that have led them to be depressed, which can definitely be helpful. So there is a hot debate, and probably truth lies somewhere in the middle, in none of the extremes. But I think that at the moment, nobody's arguing that it's absolutely either or in the field. Okay, so let's go into a more like mechanistic view of how these drugs are working in the brain. And the most simplistic model of psychedelic action has for long been proposed to be through the serotonin 2A receptor. Agonism means that they act as activators of this receptor. So this may happen at different points in the brain. And then there's like a cascade that will produce the hallucinations and also proposedly the plasticity effects. But one thing that many times is overlooked is that psychedelics are actually famous for being very, very promiscuous drugs. They have very complex polypharmacological profiles, which means that they do not only interact with a single receptor, as this serotonin 2A receptor, that is usually the one that has gathered more attention in the field, but actually, as you can see, for example, here for LSD, there is a long list of known targets that have been shown to interact with these drugs. So these drugs are far away from being selective for a single receptor, and this we have to keep in mind when interpreting the data that we are getting when using such compounds. And here I also want to highlight that one thing that these drugs have in common with conventional antidepressants and rapid-acting antidepressants like ketamine is that in their mechanism of action, BDNF and its receptor TrkB are again a very important player in their effects. This in the canonical, the classical model of psychedelic action is also proposed to be indirect, but I hope to convince you today of otherwise. But let's not get ahead of ourselves yet. So there was this paper published in 2018 by the laboratory of David Olson and colleagues at the University of California, Davis, that showed that psychedelics promote structural and functional plasticity, effects that are very similar to those of ketamine, which we have already discussed, that can induce plasticity and is a rapid-acting antidepressant. And what they proposed in this initial landmark paper was that both the serotonin 2A receptor and TrkB would play a role in terms of increased spinogenesis, so increase in these buttons that the dendritic shafts, these branches of the neurons have that are important for connecting between them and forming neural networks that then underlie behavior, and also in neurogenesis itself, that is getting more branches. So as you can see here, different psychedelics such as LSD or DMT will increase the number of spines, so potentially increase of connections between neurons acutely. And then when, for example, the LSD, the psychedelic drug, was combined with a treatment, what we call an antagonist, that prevents the activation of TrkB, these plasticity effects were abolished. So this already indicated back in 2018 that TrkB was important in the context of psychedelic treatment for induction of plasticity. More recently, this same group and other collaborators have shown that a hallucinogenic or proposedly non-hallucinogenic psychedelic analog, more specifically of ibogaine, which is not technically a psychedelic in the conventional sense that I defined today because it doesn't activate the serotonin 2A receptor. But in any case, it's a hallucinogenic drug in humans. What basically this lab did was that they took ibogaine, which is a psychedelic, changed the chemical structure, and made it in a way so that it would not trigger the hallucinogenic response in mice. And this is somewhat a complex thing to discuss. I will just very basically summarize that it is very challenging to know if a mouse is hallucinating or not when we deliver a psychedelic, because believe me, we have tried many times asking, but well, they do not reply, unfortunately. So what we do is that we have observed for long that many psychedelics that we know are psychedelic in humans do induce what is defined as a head twitch response in mice. This head twitch response consists in a very fast movement that the mice do with their heads when they are delivered dose of, for example, LSD or psilocybin. And they do that very, very fast and very soon after we have delivered the drug. So that is the way that we approach psychedelic-like effects in mice. What this lab did in this important research work was that with this analog of ibogaine that they called TBG, tavernantalog, they saw that despite not inducing the head twitch response that one would expect from a classical serotonergic psychedelic drug, it did still keep potential to induce plasticity, at least like, for example, here, based on counting of spine density, so increase of these connections. And this was actually somehow similar to what we see with ketamine. So in principle, what we can guess or suggest from the data presented in this paper is that it is possible to dissociate the hallucinogenic effects of psychedelics from their plasticity-inducing and also therapeutic effects of these drugs. It's a very interesting paper. I recommend that if you haven't checked it out and you're interested in the topic, definitely this is an important one. Okay, so more about the mechanism. Psilocybin has also been shown to promote plasticity and antidepressant-like effects. However, there is a hot debate as well on whether the serotonin 2A receptor, which is responsible for the hallucinations, is required in the mechanism to induce plasticity and the antidepressant effects. And there is some data that points out that it may be required, and there is some data that points out that it may not be required. And here I'm presenting to you, for example, some evidence that it is not required. For example, in this marker of plasticity in the hippocampus, that is the AMPA-NMDA ratio, then we see that when we block the serotonin 2A receptor with an antagonist of the 2A receptor that is called ketanserin, that is written as KET in here, then the effects, this antidepressant-like effect that the psilocybin induces, remains intact. Something similar happened. Sorry, because I have to change here. I have the view of the audience. So here, what we could see also is that psilocybin has an antidepressant-like effect in terms of an anti-anhedonic effect. Anhedonia is a classical marker of depression. That is, that things that we used to like, we do not like anymore. And mice really like drinking sucrose because it's sugary, and who doesn't like that? So whenever they are depressed, they drink less sucrose. But when you deliver psilocybin, then they go back to liking sucrose again, which suggests an antidepressant-like effect of psilocybin on these depressed mice. But when we added this blocker of the serotonin 2A receptor, what the experimenter saw was that the antidepressant effect was intact. And this was despite the animal not showing head-twitch response, which suggests a successful blockade of the 2A receptor. Then there was another interesting paper published in the same year that showed pretty much the same, but on the plasticity side, that is this ketanserin, the blocker of the 2A receptor, did not prevent the plasticity-inducing effects of psilocybin. So psilocybin still managed to increase some of the markers of plasticity, such as increase in spine head width and formation rate, which suggests that indeed the serotonin 2A receptor responsible for hallucinations may not be required for the plasticity-inducing effects and their associated antidepressant-like effects on behavior. And now you will be wondering, okay, that's all fine, great that you see that the mice seem to respond to these antidepressant, these psychedelics, and so this antidepressant effect regardless of the hallucinogenic effects of psychedelics. But what about humans? So there's a very interesting case report. So this is important to highlight. It's a case report. It's an N01. It's just a one-person report that was just very recently published, but it's very, very interesting because this is something that has not really been attempted before. And in this case, it was a depressed patient that was a 45-year-old male with a very long history of treatment-resistant depression, so was not responding to conventional treatments of depression. He was delivered a single psilocybin dose, but he was under the treatment of a serotonin 2A antagonist that would block the 2A receptor. So this patient was very upset in the beginning that despite he had received psilocybin, he didn't have any hallucinations because the 2A receptor was blocked. However, what was remarkable and what really surprised the researchers in this work was that this patient still displayed a very rapid antidepressant effect as measured by a drop in the symptoms of depression in this MADRS scale that was not only fast, but also very enduring. So this case report, despite these very early days, is very promising in the sense that it gives light into what we have just discussed about mice, that it may be possible in humans as well to dissociate the hallucinogenic effects and the 2A activation from the therapeutic and plasticity-inducing effects. Okay, so I want to just give you a small summary of what we have discussed so far. And first would be that psychedelics induce neuroplasticity through a mechanism that is shared with other antidepressants, both conventional and rapid-acting, where BDNF and TrkB signaling is required and invariably at the center of those mechanisms of action. Then there is also that therapeutic and neuroplasticity effects of psychedelics can be detached from their hallucinogenic action in mice and apparently, potentially, maybe also in humans. And finally, that psychedelic-induced neuroplasticity can be detached from the serotonin 2A receptor activity, the hallucinogenic-inducing receptor for psychedelics. Okay. So then, what are the mechanisms that are driving these common therapeutic effects of antidepressants and psychedelics and also their plasticity effect? So here we did our own contribution to the field after long years of work by excellent, brilliant scientists. You will see some of them in the upper right corner of the screen. Unfortunately, I don't have time to introduce the marvelous work of all of them, but please bear with me, otherwise we'll be here for much longer than I assume we all have time for. But basically, what we have been working on is trying to dissect what is the mechanism through which psychedelics promote plasticity and induce these therapeutic effects. Of course, our work, also I want to highlight, are done in vitro and with animals. We have not worked with humans, so everything that I will say so far applies to mice in vitro, and we'll see if it also works in humans. We don't know yet. Okay, so first of all, what we have now discovered is that psychedelics also do bind to TrkB, but in this case with much higher affinities than the antidepressants, conventional antidepressants would. In here, what you can see is on the right side of the screen is a very wonderful atomistic model that we developed in collaboration with the group of Ilpo Vatulainen, guided by Misa Giris and Gira Enkavi, marvelous computational physicists that are experts in biomembranes and receptor proteins. What they basically did was take TrkB, they put it in a physiologically relevant membrane environment, and then atom by atom, they simulate what happens and what changes are induced by LSD. And what they found was that LSD, that you can see in purple here, very fastly goes into the transmembrane domain of TrkB. So this is the membrane of neurons, and then it binds to this side. That would be the extracellular facing crevice of TrkB dimers. And what it does through doing that is that it stabilizes the dimer and it stays there for a long time. Of course, these are very detailed simulations that we can do thanks to the super computers that we have available at the University of Helsinki, and also in Finland. Actually, we use Lumi, which is one of the most powerful supercomputers ever invented, and they have given us a very clear picture into what may be happening at an atomic level. But of course, we were not just happy with using supercomputers for some interesting molecular dynamic simulations. We went into the wet lab and we looked at what was happening with actual proteins and drugs. And for that, what we did was using radioactive LSD, so radio-labeled LSD, what you can see here. We checked the interaction directly with the TrkB receptors, and what we found that was that they interact at very, very low concentrations, which do indicate this is a very strong interaction, a very high affinity interaction. However, when we introduce some point mutations into some of the residues, these small pieces that proteins are built from, then we made some small changes that we thought could disrupt the interaction with the drugs. And we found that indeed, when we did those very small changes, we abolished the interaction with psychedelics, indicating that there are very specific interactions that are important to stabilize these TrkB LSD bound states. We also did look at how long these drugs stay inside the TrkB, and what we found was that they stay for a remarkably long time, for several hours. In fact, that means that the interaction is so strong that it takes quite a lot of time until the receptor dissociates from LSD by themselves. Remarkably, also, when we introduced this point mutation that would diminish the interaction between LSD and TrkB, we found that this interaction indeed very fastly dropped. So this indicates that this was a very important residue to stabilize the interaction. Finally, we also wanted to see in live cells whether TrkB monomers, which are inactive, would dimerize and for how long when treated with LSD. And what we saw was that when TrkB is in the membrane, they are like monomers. So what you would have is basically one of these alpha helixes, okay, by themselves. They are not forming a cross. They are just sticks across the membrane. So when we add LSD in live cells, then what we found was that the dimers go and dimerize and adopt what would be their active conformation, forming this dimer. And this happens also pretty fast, takes around 1 hour, and it takes quite a long time until the situation goes back to normal. Again, when we introduced this point mutation, we abolished this particular effect. Okay, then we were not happy still with that, so we wanted to be very sure about what we got. And among other orthogonal methods that we used to look at pretty much the same, that was whether these drugs were binding to TrkB, we attempted many kind of structural biology approaches to this, but The problem with TrkB that we have is that the pocket, the binding site for these drugs exists in a very kind of a difficult to study portion of the protein. That would be the transmembrane. And not only that, we also need a very specific kind of conformation for it to be able to interact properly with psychedelics. So after really fighting very hard, we got like some amazing laboratories joining into the effort, and through NMR, which NMR spectroscopy, which is a structural biology technique, we went and looked whether what we were seeing in the simulation at atomic level would be reproducible in this NMR, this actual protein versus LSD situation. And what we found was that indeed, what we see is that when we add LSD to TrkB inside lipid bicells, cells, where we have like TrkB embedded, then we observe that the kind of what's called the CSPs, or chemical shift perturbations, happen mainly in the dimer at the site that we were expecting from our mutagenesis studies and our computational dynamic molecular simulations. That's this extracellular facing domain of TrkB dimer. So just about here, which in our mind clearly confirm that indeed there is an interaction that is happening at that place involving many of the residues that we were expecting to interact. And among them, it was Y433, which is Y434, because for this we used human TrkB. We again found that this is a crucial residue to stabilize the interaction with TrkB, as it was one of the residues that received the most, the highest perturbations in the NMR spectroscopy, indicating that there was an important conformational change taking place. Okay, so TrkB binding of psychedelics confirmed. Then we also wanted to get an idea of how would they compare to conventional antidepressants, of course. And for that, what we did was competition binding assays, where we had like LSD or psilocin, that you will see again as PSI, okay, Psi. So this is psilocin, the active metabolite of psilocybin, and then fluoxetine, a conventional antidepressant. And what basically you can see in this graph here is that while LSD and psilocin have a very low Ki, which indicates that there is a very high affinity for TrkB because they displace the radiolabeled LSD at low concentrations, this is not the case for fluoxetine, that requires micromolar range of concentrations to displace radiolabeled LSD from TrkB. In other words, it takes a lot more fluoxetine to kick out the same amount of LSD from TrkB, which indicates that fluoxetine, as we had previously published, is not a very high affinity interactor. So we also simulated this with our computational physicist colleagues, how this kind of conformational changes may differ. And what we found was that the binding sites are actually different, despite at some points they seem partially overlapping. And the conformational shifts that they induce are actually different, which may actually underlie the differences in affinity, and also potentially the differences between the rapid-acting and persisting antidepressant effects of psychedelics when compared to conventional antidepressants such as fluoxetine and even ketamine, which is another low affinity binder of TrkB. Right. So psychedelics also do stabilize TrkB dimers, as we have seen before, and we have done several super detailed computational molecular dynamic simulations that show us that when we have a level of cholesterol in the membrane that resembles that of active synapses in the adult brain, so the ones that we actually would be targeting when we are treating patients in principle, we see that TrkB dimers adopt an inactive conformation that is basically the two monomers are in parallel in the membrane. So this is an inactive conformation, and it's quickly internalized and degraded by the cell. However, when we introduce psilocin or LSD into the system, despite the high cholesterol that usually would drive this inactive parallel conformation, they act as some kind of wedge that keeps the TrkB dimer in a cross-shape conformation that is associated with their active states. And this is what we believe will drive the prolonged neurotrophic signaling that brings about plasticity and also if everything is fine, the antidepressant effects. So here what we see again is that when we introduce the point mutation that abolishes TrkB interaction with psychedelics, this dimerization effect that is concentration dependently, as you can see here for psilocin and for LSD, is completely abolished, which then again highlights how important this interaction is. Then about how these psychedelics interact with TrkB and what is the role of the endogenous ligand of TrkB. So that is BDNF. We all have the BDNF. In fact, it stands for brain-derived neurotrophic factor. We all have BDNF right now being released into our synapses, as we learn with this presentation, hopefully. And this, what we propose and what we have actually observed from our experiments, is that psychedelics will not activate TrkB by themselves. They are not agonists, as they are, for example, for the serotonin 2A receptor. They are positive allosteric modulators, which means that what they do is that by binding to TrkB, they promote that the active conformation induced by BDNF remains active for longer. So this is what they do. They do not activate TrkB on themselves. They promote that BDNF will produce an activation that is more long-lasting, perhaps more pronounced. Okay, and this we observe because when we basically take out BDNF from the environment, from the extracellular environment, then all the effects of LSD are abolished, as you see here in terms of TrkB dimerization, which highlights that BDNF is an important player that must still be taken into account when interpreting this kind of psychedelic induced effects. The model that we propose is that the inactive synapses where BDNF is released, then LSD or other psychedelics will facilitate the active conformation of TrkB, which then brings about the plasticity and survival of that particular synapse. Because BDNF is released in response to activity, and activity can be produced, for example, by psychotherapy in terms of the patient, so that you can start to relate it to what happens perhaps in the clinic. However, when there is no activity in that particular synapse, even because, for example, in that circuitry, there is not much activity going on while the psychedelic is there, LSD will not be able to facilitate BDNF signaling because there is no BDNF in the extracellular environment. Therefore, there will be no neurotrophic signaling, and that synapse may just like stay there as it is, or even retract and disappear. So this is the way that we think that BDNF is going to be crucial to modulate circuitry in the context of psychedelic treatment. So we also have observed that through a very detailed mechanistic set of experiments, that psychedelics can increase TrkB trafficking into the cell surface, and you can see here that already in vitro with neurons in a dish, they can increase the expression, the localization of TrkB into the membrane, the plasma membrane of the cell, which is where it will be able to access BDNF. And this actually is an effect that remains for quite a long time, facilitating, therefore, that BDNF will prolong its neurotrophic signaling. And this is an effect that is independent from the serotonin 2A receptor, as observed by that when we pre-treat the neurons with an antagonist, a blocker of the 2A receptor, this effect is still untouched. And you're going to be seeing this a lot in the rest of my slides. Then what we also observed is that psychedelics like LSD and psilocin will rapidly promote that TrkB is trafficked into dendritic spines. That is, again, where in principle in the brain, your neurons will make connections with each other. So TrkB is needed there, and what the psychedelics are doing is that they are pushing TrkB to be where it is required so that it can be activated by BDNF and then induce plasticity. However, when we abolish this kind of interaction, LSD and psilocin lost this effect of being able to push TrkB to be in the site where it should be to be activated by BDNF. We also have seen that psychedelics do promote plasticity through a specific TrkB signaling pathways, so downstream of the activation of TrkB through BDNF and facilitation by psychedelics, you will have different molecules that will take into the intracellular side of the cell a cascade that will then trigger the neuroplastic effects that we see at the neuronal level and then at the neuronal network level. And what we have observed is that, for example, LSD in neurons will increase the phosphorylation of ERK, which is a very important intracellular signaling pathway for survival and neuroplasticity. And they do that with a very rapid effect. You see already at 5 minutes, there is an upregulation of phosphorylation of ERK that remains at 1 hour, but this is gone already at 24 hours after the treatment with the LSD. Again, when we try to block this serotonin 2A receptor with the antagonist, we see that these effects remain, and therefore it's not surprising that all of the plasticity and antidepressant effects would also remain, because TrkB is being activated regardless of the 2A receptor activation that brings about the hallucinations. So we also have observed that another important interaction of TrkB with another intracellular signaling molecule, that is PLC gamma 1, is increased. And here in these wonderful pictures, what basically you can see is every single dot is a molecule of either TrkB in blue or PLC gamma 1 in yellow. And you see that what psychedelics are basically doing is that they upregulate, they increase this kind of interaction, which then is, as we have seen in this previous diagram, also brings about here to PLC gamma plasticity downstream of TrkB and BDNF activation. So one more evidence that TrkB is crucial. Here we were, of course, very interested as well in looking at proper neuroplasticity at the structural level, so how these psychedelics can induce spinogenesis or dendritogenesis. And what we see is that while psychedelics successfully increase spinogenesis in what we call wild type or normal neurons, when we introduce this point mutation that abolishes the interaction of TrkB with psychedelics, then these effects are completely blocked, suggesting that these plasticity-inducing effects of psychedelics do require that TrkB can interact with psychedelics. So it is a key target. Then when we looked at what happens if we take BDNF out of the equation, that would be with this TrkB.Fc, the blue column, we see that without BDNF, the effects on plasticity are also abolished. This is not surprising. It's because a positive allosteric modulator of TrkB will require BDNF for its effect. So this is what psychedelics are. They do require BDNF. And then, again, when we block the serotonin 2A receptor with the selective serotonin 2A receptor antagonist, we still see the plasticity effect, which in our mind further adds to the evidence that the 2A receptor is not required for the plasticity-inducing effects of psychedelics. Then when we go to look at dendritogenesis, so this neuronal branching, pretty much we see the same. So LSD and psilocin will increase this dendritogenesis, increase the number of branches, increase in the possibilities that neurons have of communicating with each other and reshaping neuronal networks. But with the mutation that prevents interaction with LSD or psilocin, these effects are abolished. Then again, mimicking what we saw for spinogenesis, blocking of the serotonin 2A receptor does pretty much nothing, so plasticity effects still induce. But when we take out BDNF, then the plasticity effects are abolished. So BDNF, again, important in the equation. Now let's look at some behavior, and for that, what we have used is different animal behavioral models of depression and others. Particularly for antidepressant-like effects of these drugs, what we have used is this forced swimming test kind of model, where we submit the animals to induce a depression-like behavior. For several weeks, the animals are put to swim in a cylinder where they cannot escape, so they obviously end up getting a bit frustrated and potentially depressed as a mouse could be, I guess. And then they stop swimming, which is interpreted as that the mice are more depressed, right? Then what we do is that we get that baseline, we treat the animals with either vehicle or the serotonin 2A antagonist, the 700, 907, that will block 2A, and then we deliver 30 minutes later either a dose of saline, which is our control, or LSD, okay? Then we put the animals back into their home cage, and they are left undisturbed for 7 days until we go back and do a forced swimming test 7 days later. And what we have observed was that when we block the head twitch response, so this hallucinogenic-like effect with the antagonist, of the 2A receptor, the antidepressant-like effect of LSD remains intact. So 2A does not seem to be involved in this antidepressant-like effect in mice. When we looked at the head-twitch response, which again is this kind of behavioral proxy for hallucinations in mice, okay? So what we see is that LSD in animals that are, just like given LSD will trigger this hallucinogenic-like response. You see that it skyrockets to more than 30 twitches, total counts in 30 minutes. But when we do pre-treat the animals with the 2A antagonist, this effect is blocked. This basically is not a new discovery. It was widely known from before our work that serotonin 2A receptor drives the hallucinogenic response both in mice and in humans, and that this kind of antagonist treatments will abolish the hallucinogenic response both in mice and in humans. And indeed, this is the case. So in here, what we can take from this data is that the 2A receptor is not involved with the antidepressant effect, but it is involved in driving the hallucinogenic effects of LSD. Right, but what about TrkB? So for that, what we did was that we developed a mouse that includes this point mutation in TrkB. Okay, it's a heterozygous mouse, so it means that half of the TrkB will be normal and the other half will include a mutation. The thing is that when these two combine, the heterodimer that is called of one mutant and one normal, one wild type, a monomer of TrkB, this will have the effect that despite they are otherwise normal when compared to their completely like wild type, so normal TrkB with normal TrkB animals, they will not respond to LSD treatment. So what you see here is that LSD, again, in normal animals will induce this antidepressant-like response, but in these mutant animals, it will not. It will not. So this means that TrkB interaction is required for the antidepressant-like effect of psychedelics. However, when we go and look into the head-twitch response, what we see is that our mutant animals that did not respond to LSD and did not show any antidepressant-like effect in response to LSD, they do show a completely normal head-twitch response when compared with their wild-type determinants, so their normal siblings. And this indicates that TrkB interaction with psychedelics is not involved in this kind of hallucinogenic-like effect in mice, and potentially comes to suggest that in humans, it may be possible also to dissociate the effects that psychedelics have on TrkB, that would be plasticity-inducing and antidepressant effects, from the hallucinogenic effects through the serotonin 2A receptor. Okay, so not happy with that. We also use some other behavioral models, like this fear extinction model. I will not go too much into detail, but you can get the idea that it's meant to mimic what happens in post-traumatic stress disorder. So there, what we wanted to look at was whether animals that had an association of a given context, a given environment with fear, did become like fearful, obviously, of that environment. And then what we do is that we treat the animals with the LSD or saline, that is our control. Okay, so those animals do not receive LSD. And then we submit them to 3 extinction training sessions. That basically means we put the animal back into the same context that they were afraid of, but the reason why they were afraid of is no longer present. So at first, they are obviously very afraid because they, like in PTSD patients, they will associate that environment to the fear. But what we saw was that in this very short term, at the end of the extinction session, number 3, we saw that LSD in normal wild-type animals had facilitated this extinction training. So basically, the animals that received LSD and were genetically normal, with normal TrkB, respond like they would to an antidepressant, but with a single dose of LSD instead of several weeks of treatment with a conventional antidepressant. However, when we went and checked our TrkB mutant animals with LSD, what we saw was that in these animals, LSD was not able to facilitate the extinction training, this kind of psychology, like psychotherapy for mice kind of effect. Okay? Then this was in the short term, so just a few days after this single dose of LSD, but we were also interested in the long term because this is something that we all are interested in when looking at the psychedelics when compared to other antidepressants. That is not only that they are faster, but also that with a single dose, they seem to have a much longer lasting, much more persistent antidepressant effect than chronic treatment with antidepressants that usually require for the patients to continue the treatment for months, even years, in some cases, even for the rest of their lives. So this is something that psychedelics have brought attention to. So we also wanted to look at this longer time point. And then 28 days after we had delivered this dose of LSD to the animals, we go and what we see is that without any more treatment, the animals remained less afraid from that environment that initially they associated with the fear response than the mutant animals that also had received LSD. Therefore, this further adds evidence that TrkB interaction is required for all of these antidepressant-like effects in behavior of psychedelics. Right, so I'm almost finishing now. Another kind of experiment that we wanted to do is looking at neural network plasticity. And for that, there is a gold standard in the field of plasticity in neuroscience that is this ocular dominance plasticity model. So basically what we do with this model is that we induce amblyopia in adult animals. What we do is basically we turn our animals into some kind of parrots. We put a patch in one of their eyes, and this is called monocular deprivation. So we do that for a whole week. And what we do is that during that week, we administer 3 different doses of LSD every 72 hours. Okay. Then what we do is that we remove the patch from that eye from the animal, and then just let the animal, do its own life and basically see the world with both eyes. And what we have observed is that LSD does induce a shift in the ocular dominance index, which means that it's making the neural networks so plastic in the adult brain when usually they would not be that plastic in the visual cortex, that the animal turns amblyopic. This is something that only happens during childhood. During childhood in humans, you may have heard of the lazy eye. That's what clinically is called amblyopia. So what in this kind of context is done is that the good eye is covered with a patch so that the lazy eye kind of can work more. And then at the visual cortex level, that is where the brain interprets the visual signals, you are forcing that both eyes have the same importance, the same weight. Because otherwise, if you don't treat amblyopia during childhood, once the patients go into adulthood, the neural networks become so rigid that you cannot change them anymore, even if you add the patch like a child. Because as we grow, and this is very intuitive, I think, for many of us, our capability to learn is decreased. And this is because there is, in a way, less plasticity in many of the circuitry that drive our behavior and our learning capabilities. So what we did with this animal was basically take the opposite, that is, take an adult that is completely normal, and then add the patch and induce amblyopia. Okay, and then because LSD was capable, together with this monocular deprivation, to induce an ocular shift, we interpret that psychedelics can induce a state of plasticity enough in the visual cortex that resembles that one of early stages of development, of childhood, if you will, for the mice. Okay, so this for us is a clear marker that these drugs are capable of inducing neural network plasticity in a generalized manner in the human brain. We also checked at the long-term survival of neurons in the hippocampus. That is an important area of your brain to kind of like get for memory, basically, and for consolidation of memories later on. And what is being proposed for a long time is that there are neurons in the hippocampus that are kind of being born all the time. Not many, but they are being born during adulthood. This is one of the only regions that have been shown, at least in mice and in some studies even in humans, to be able to produce more neurons during adulthood, because most of the brain, the neurons that you are born with, those are the ones that are going to come with you to the grave, pretty much, because there is no neuroregeneration. So the hippocampus, the ventral gyrus in particular, that is where we were looking at, is one of the only regions where you will see these newborn neurons even in adulthood. So for that, what we did in this experiment was that we tagged these newborn neurons with something that is called BRDU, that basically will show us which neurons were being born at this particular time in day one. The next day, we delivered a single dose of either saline, the control, or LSD to the animals. Then left the animals undisturbed for 1 month, pretty much up to day 28. And then we sacrificed the animals and we looked at what happens to these newborn neurons. Then what we observed was that LSD, similar to actually chronic treatment with antidepressants, was capable of inducing an increase in the number of newborn neurons that survived after 1 month. So in a way, you are aiding these neurons to, mature and get included into the network. So it's in a way a marker of neuronal survival and plasticity and neuronal network plasticity. While the animals that had the TrkB that cannot interact with psychedelics saw this effect abolished, which we interpreted again as the TrkB was crucial to mediate these kind of effects. And given that the hippocampus is so important for memory in general and also has been involved in depression and other mood disorders, we think this is also an important finding to take into account. Okay, altogether, I want to propose to you a mechanistic model for the TrkB-mediated psychedelic-induced plasticity effects that may be also associated with the antidepressant effects themselves. First of all, we have TrkB that will be monomers in the membrane, and when we increase the amount of cholesterol, the concentration of cholesterol in the membrane, which happens, for example, in the active site of the synapse where TrkB should be making its effect, then TrkB will naturally, without psychedelics, get this parallel conformation that is inactive. So there is no signaling, does not induce plasticity. When we do have BDNF and LSD, even if there is this high concentration of cholesterol that usually would inactivate TrkB, what we get is that LSD or other psychedelics can act as a wedge that stabilizes the TrkB cross-shaped dimer conformation that then brings about its activation and neurotrophic signaling that then leads to plasticity and antidepressant effects. This is important to understand that these drugs as positive allosteric modulators, they do not activate TrkB by themselves. They will require BDNF. And BDNF is only going to be released in response to neuronal activity. So once we get an active synapse, LSD is going to facilitate this plasticity and survival effects in that particular synapse or in that particular neuron. However, if there is no activity, there will be no signaling and potentially retraction of the cell. So that is the way that at synaptic level, we propose that the changes may be being induced at neuronal networks as well. So through driving activity, for example, with psychotherapy in humans. Then what these drugs will do is that they enhance plasticity in a generalized fashion as long as there is activity. But then there will be an activity-dependent pruning that will finally shape what kind of changes our neuronal networks would have in response to psychedelic treatment and environmental factors. There is also an increase in the survival of newborn neurons in the hippocampus, which then again is important in the context of mood disorders and memory that may be associated also with their behavioral effects, because this in a way also contributes to neural network plasticity in the hippocampus circuitry. Finally, at a more neural network level and the behavior that they are underlying, what we propose is that through this increase in neural network variability induced by plasticity, then it matters what we do while this plasticity window is reopened. So that we are going to be able to drive mainly through environmental interventions. In our case, what I showed you today was this kind of like extinction training, this psychotherapy for mice, or the patching of the eye in the amblyopia ocular dominance plasticity experiments. This is what really is going to drive the activity and then induce the specific changes in neural networks. And the consequence of this, we believe, is that psychedelics on themselves, despite they may induce plasticity, they do not necessarily produce antidepressant effects. Because what will really drive the outcome, the behavioral outcome the plasticity of the psychedelic treatment will be what kind of environment we are in, or what has been for long known in the environments that are associated with psychedelic use, the set and setting. So all in all, that's what's the environment. So the environment is going to be critical, and that's why we think that in modern clinical trials, we're seeing a lot of positive effects with psychedelic-assisted psychotherapy, psychotherapy being a key here. And this, we also believe, is why there is like anecdotal evidence, and probably many of you will have heard about, that psychedelics used in a recreational setting are not necessarily therapeutic. In fact, they can be the opposite. There are people that may end up with, PTSD-like symptoms from what's called very bad sleep that is not properly like integrated and talked about. So that we think is explained by the fact that these drugs are just unspecific amplifiers. So they induce plasticity, and whatever you feed through that open window is what will drive the final change. Okay, so with that, I want to give you with an even like simpler summary that you can take home, especially if I bored you with all of these technical details. I'm sorry I had to do it, and I'm academic. I'm a scientist, and that's my job. They pay me to do that. But in very, very like summarized form, well, we can say that psychedelics mainly will have two paths. One is the serotonin 2A path, serotonin 2A receptor path. There's the one that drives the hallucinogenic effects that psychedelics are well known. And then there's this novel path that we have now uncovered, that is the TrkB pathway, that is also shared with other antidepressants, despite psychedelics seem to do it much better. Where psychedelics like LSD will bind at the TrkB transmembrane dimer domain, and then facilitate its activation by BDNF and induction of plasticity. Then this TrkB dependent enhanced plasticity that can be measured, for instance, by increasing spinogenesis and dendritogenesis, is what, together with the environmental interventions, will bring about the neural network rewiring and the antidepressant effects if the environment is right. And finally, I want to leave you with a message that is that we believe that all of this may actually explain that the psychedelics having a higher affinity for TrkB could potentially explain their faster and more persistent antidepressant effects in the clinic of psychedelics when compared to other conventional antidepressants that require several weeks of daily treatment to just start seeing some effects on more. So with that, thank you very much, first of all, to my co-authors, collaborators, and other people that have made this possible. We are just 33 people in the author list of this particular paper, but I can warrant you that there were a lot more people than that making all of this research possible. Some of them are included here, some other, unfortunately, I didn't have a space for them. But I want to bring attention particularly to Eero Castrén, that has been my mentor and PhD supervisor, together with Plinio Casarotto, that was the postdoc I was working with for my master's and also during the first part of my PhD. And have been critical for the success of this project, and also especially our collaborators in the Department of Physics at the University of Helsinki, Ilpo Vatulainen, Misha Girish, and Girai Enkavi, together with the members of the Eero Castrén lab, like Cecilia Brunello or Caroline Bioggione, Merve Fred, and many others like Lauriel, Sila, Sven Saxon, and so many other people that really make this possible. So thank you very much to them. Also, thank you for my funding during the PhD that was granted by the doctoral school in Brain and Mind, and all the other funding agencies that made this research possible. So with that, Kitos, thank you very much for your attention. I hope not to have bored you too much, and I'm happy to take any questions and start a lively discussion with you all. Thank you.
Thank you, Rafael. That was amazing. Yeah, so let's go straight to the questions. Unfortunately, we have only a little more than 10 minutes for the questions because of, well, the technical difficulties and Rafael's timetable after this. So everyone, you can post your questions in the Q&A part that's in the Zoom panel. So not in the chat, but in the Q&A. And if you don't want to read your question out loud, please mention that at the end of your question. And you can also upvote the already existing questions. And here we have Ville's question. as first. Would you like to ask it?
Yes. So congrats on the excellent work and presentation. So I wonder, because there are like reasons to think there could be endogenous psychedelics due to the similar experiential content, but do you think there is an endogenous psychedelic activating this pathway that you presented? As I understand it, I'm referring to the Vargas paper that suggested that this 5-HT2A intracellular signaling could not be activated by serotonin as it does not enter cells. But this BDNF TrkB is an extracellular pathway. So could it be serotonin or could it be something else? Like what is the function of this modulation without something from the outside? I also understood that you say that this serotonin 2A intracellular signaling is not necessary for promoting neuroplasticity, which seems to be in a little bit of conflict with the Vargas paper, as I summarily understand it. Thank you.
Okay, thank you, Ville. Those are very good questions. So I will start with the first one. First of all, clarify that this is purely a speculation because I don't have any data to back this up. Like it's difficult also to kind of reconcile with the idea of an endogenous psychedelic, right? Because I don't know if there was an endogenous psychedelic, would it mean that we'll be having like hallucinations every now and then? Some people may say that dreaming is some kind of hallucination in a particular time of the day. But yeah, it's a bit of a complicated question here. My kind of guess here would be that there's BDNF. I mean, that's for sure that it is going to activate TrkB, and we know that it is crucial for TrkB activation. Whether there is some other endogenous ligand that would activate TrkB in a similar fashion, I understand that's what you propose. As psychedelics that would promote the activation of TrkB. The simple answer is that we just don't know if there is anything. Like we have particularly never heard about anything that would do that we know. But this is a novel binding site, and this is the very interesting, and really what has got us so excited about this finding, that it's a completely novel binding site that nobody had reported before. We are the first ones to see it, and I understand that because it's a very challenging binding site. It's not something easy that you just stumble upon. So yeah, I would say that we just don't know. We just don't know. Then regarding the Vargas paper from Olson's lab that pointed out that 2A intracellular pool being important, I think they really made a good point that the 2A receptor is important for the hallucinogenic response. They clearly seem to show that for the membrane 2A receptor. Whether the intracellular pool of 2A is also required for plasticity, I think remains an open question. It is true that, I really respect all of the work that is done in David Olson's lab and collaborators. I've been an admirer actually for a very long time of their work. But it's also true that this is science and we find contradictory data all the time, right? And the data that we have is that when we use an antagonist of the 2A receptor, we do not see any kind of problem like what we see still plasticity induced by psychedelics. So this to us points out that the 2A receptor, whether intracellular or not, does not seem to play a role in the plasticity inducing an antidepressant-like response. But it's an exciting open question, and this is just early days. So I think that there is a lot of open questions and unknowns that are still there, and we just have to accept that.
Oh, thank you very much.
Thank you. And then we have Hannu's question. Hannu, would you like to open your mic and ask it?
Okay, is it okay now? Can you hear me? Okay, good. Thank you. Excellent presentation, very scientific. Congratulations also with the very, very interesting data. Well, before I make my question, I just a little bit give a background why I do this question. I'm the primary investigator of a clinical study here in Finland and in Canada, psilocybin assisted treatment on the treatment of alcohol dependency. We have now 140 patients already, and we will see how it goes. But my question is, because it's very commonly assumed that the hallucinogenic effect is required for the therapeutic effect. Your data shows kind of opposite. But my question is there any human data to support this assumption that the psychedelic effect is required?
Sorry, Hannah, for interrupting with that. Yeah, thank you very much. This is a great question, and actually, I think it's really, a very important point in the field. There is actually no very strong data that would suggest that this is the case. There is certainly some correlative observations that, for example, mystical experiences in some of the papers that have been published so far in small studies seem to correlate, not necessarily cause, but correlate with, the antidepressant effect or how long-lasting it is, for example. But that's not always the case. There are also patients that have shown a rapid-acting and long-lasting antidepressant effect without having a mystical experience necessarily, or having a very deep, psychedelic experience that revealed what was the new perspective that would heal their lives from that thing that was before keeping them depressed, right? So there is not really definitive data on that. It's also kind of ethically complicated to design an experiment like that with humans, because of course we know that the psychotherapeutic factor is very important, and I do know that there are at least some small studies underway to check whether patients of depression that do not have some a strong timeline of psychotherapy associated with the psychedelic administration would respond to that. But in most trials, it kind of will many times not even cross the ethical committee barrier if you do not include psychotherapy, because we know it's so important, right? Like the set and setting, and the users of psychedelics know it very well. So I do not think that there is definitive answers to that. Having said that, there is yet not evidence of the opposite that is very solid either. Like I showed in my presentation, a first case report that was a first in class, because this is also very recent. This was not a question before. Before it was kind of assumed that the hallucinogenic effect would be required. But this patient that did show a rapid-acting and long-lasting antidepressant effect with a single dose of psilocybin and no hallucination, including the 2A antagonist, that indicates that this may be possible. It may be possible. So it's a very important question, Hanwan. I'm sorry that I don't have a definitive answer, but at least this is the current state of affairs as far as I'm aware.
Yeah. Well, yeah, I agree with you. I think no one has yet the answer for that. But we had to remember that in the case reports or uncontrolled studies, we must be very critical when we do any kind of conclusions.
Absolutely.
And so far, we don't have any controlled studies to show that this is the case. So it might be a very wrong assumption, what is now kind of generally believed that this is required. But okay, let's leave this subject. And my next question is that this all your excellent studies and research is animal research. Is there yet any similar research in humans? Sorry, I have not been followed this field, but you know it, so tell me.
Well, so I mean, the TrkB findings were just published with our paper last June. So unfortunately, I don't think people have had that much time to start looking into this. There are several problems with this, that while we have some well-developed serotonin 2A antagonists that have been used in humans, like for example ketanserin, to successfully block the hallucinogenic response induced by, for example, LSD, there is not a good antagonist for TrkB that has been validated and well characterized in humans. So this makes the study of TrkB in humans a bit challenging. From the perspective, for example, of positron emission tomography, PET, that is something that is commonly studied, commonly used to study receptor occupancy in a living brain of a human. There is also not a good selective radioligand of TrkB, particularly, especially for this novel binding site, at least that we know so far. So these are things that make studying TrkB in humans very challenging at the moment. But I'm aware that there are many brilliant people working on solutions. I'm sure of that, and hopefully, I'm very excited to see what people do with this in the near future. And I'm very happy also that you finally got the green light to work with clinical trials and psilocybin in Finland. I know that this has been a challenge for quite a while, and ethical committees have been kind of pushing back for years. So it is the first news I get. So yeah, happy to hear that the research can go on.
Thank you. Maybe we will have the results in a year or so. But anyway,
Best of luck.
Thank you. And that's all. Let's leave questions for other people also.
Yeah, great. Looking forward.
We have very little time now, but there's a really interesting question. Please comment on the idea of designing a clinical psilocybin depression study where TrkB was blocked and compared to a control group with regular psilocybin treatment.
Yeah, I think that would be an amazing question, and it kind of links to the previous question that I just answered, and is that we may not have the right tools yet to work on that. I mean, that would be wonderful, and it's pretty much the same kind of study that should be done with a 2A antagonist, which should be more readily possible. I mean, this case report with the 2A antagonist kind of like shows that it is possible. So yeah, I mean, ideally, yes, I'm dying to see something like that. I'm afraid that I will not be the one carrying out that kind of experiment, but really looking forward to whomever has the tools and the resources, please, like, do it. And I look forward to see what happens. I mean, this is important. It's necessary to see whether what we are observing in neuronal dishes, like in neurons in a dish, and in mice also translates to human. This is not guaranteed. What we know is that because we have attempted that, psychedelics do bind to human TrkB in a very similar fashion as they do to the rodent TrkB, to rat and to mouse, because the region is very conserved, maybe for a reason, right? That we didn't know until now, but they are very conserved. So I would expect that this TrkB interaction with psychedelics will also happen in humans, but it remains to be seen whether we will see something that resembles what we have now observed with mice. But great question.
Thank you. Now I think it's the final question. Jussi, would you like to read it out loud?
Hi, can you guys hear me?
Yeah, perfectly.
Awesome. Hey, yeah, so I think in the beginning of your presentation, you mentioned that the psychedelics can activate the neuronal spine growth. And I know that the decrease in the spine formation is also a sign of some dementias, for example, Alzheimer's disease. So I was wondering, like, does that kind of indicate that treatments or drugs that can activate the neuroplasticity could also be used to help to treat dementias?
Great question, Jussi. Thank you. Okay, again, I want to highlight that in my response, there's only a speculation because I don't have, and I don't think at the moment there is any publication that would suggest this, but potentially, given that, as you well said, neurodegenerative disorders are characterized by loss of synapses or spines. One would expect that some kind of treatment that upregulates neurotrophic signaling and brings about, more spines could be a marker to say that you have some kind of therapeutic tool that could be psychedelics, for example. However, I am, to the best of my knowledge, not aware of a single report that suggests that psychedelics are useful either in preventing or in treating neurodegenerative disorders. That doesn't mean that it wouldn't be possible. But I would suggest that we have to be very cautious in what we say at the moment because there is no data available to make claims on that. My guess here, and again, speculation alert, is that potentially they could have a use. I'm very, very skeptical that they could be curative. We, I think, are not that sure either of what are the actual causes for most neurodegenerative disorders. So I think that upregulating plasticity may not solve the core problem, which is really what would make a curative solution, but it may be helpful in, kind of as a palliative or as a side treatment to some other like curative treatment that would actually like go and target the core problem of neurodegenerative diseases that in many cases remains to be found. So yeah, that's again my kind of disappointing, I guess, answer in many cases. But I really like to respond with data in hand. And as I don't have data in hand now, I can only have my guesses. So that there you got my guess. That's the best you can get from me right now.
Thank you. That was great.
Thank you.
Thank you, Rafael, for the amazing presentation. And thanks everyone to great questions. So now it's 1 minute until you have to go. So would you like to say some last words?
Well, I would like to thank all of you for your attention and interest. I think this is a very, very hot field at the moment. I think maybe even too much. So I would advise everyone to be cautious of what we expect psychedelics to do, because it's true that we have had them forbidden for a very long time, and that kind of has pushed us to go to the other side now that we have seen that they do have some therapeutic potential. But we should keep in mind that there are some risks associated with the use of these drugs. So with that in mind, I would just like say that let's contribute and help in the way that we can so that research can continue, but let's also be careful and not jump into conclusions and, for example, start advocating for like open use of psychedelics for everyone, because I do think that may have a very negative impact in the longer run on the research itself, as it happened already in the 60s and 70s. That led to forbidding even some kinds of research with these drugs. So all in all, there's reason for excitement, but please, let's be cautious. Let's look at the data, and we do not have all the data yet. So let researchers do their research. So thank you all.
Great, very nice points. See you later.
Thank you. Best of luck to everyone, and thank you for your great questions. Bye.
Bye-bye. So thanks for everyone for attending. Our event producer, Henry, will share now a slide of upcoming events, upcoming Psykedeelitutkimusyhdistys events. So if you're not yet a member of Psykedeelitutkimusyhdistys, we invite you to become one. You can buy a support membership in our web store at holvi.com/shop/Psykedeelitutkimusyhdistys, and you can find this information on our website too, psykedeelitutkimusyhdistys.fi. And if you are working or studying in an academic discipline that is relevant to psychedelic research, and you currently carry out or are planning to carry out psychedelic research or wish to actively advance such research, you may apply for full membership. And you will find more info and the application form at our website. And we also accept donations. More of that is also on our website. And you can follow us on Facebook, Instagram, or Twitter, or X. And so we can keep you updated about our future events and other activities. And tonight or tomorrow, we will send you a feedback form about this event, and we ask you to answer it so that we can make improvements to our events. Thank you. That was all for tonight. See you later.